So we will continue with the discussion of microbial taxonomy. In the previous video, we have seen the basic idea of classification, the three interrelated processes of classification or process of taxonomy as classification, nomenclature, and identification. And also we have seen what is a species and what are the different hierarchical levels of classification. Now we are moving into the classification systems. So there are different classification systems used in living organism for the actual classification, right? So in which it is like there will be certain intuitive type of classification. That's what we call the natural classification, in which the very basic natural features, that means the biological features of organisms are used, or these characters are used for inclusion of a particular organism into a group, right? So this classification arranges organisms into groups, and this group has a property that the members share many characteristics and reflects as much as possible the biological nature of organism, right? That means most of the observable and physical properties are similar in a particular group, right? This is what we call the natural system, or it's a kind of intuitive system, right? You can see examples like when we classify human beings, we have a common feature like mammals usually will have long hair, or they will have hair, then self-regulatory body temperature, milk producing mammary glands, etc. in female. So these are some common features, and based on these common features we include, and also there is the physical appearance and other features. So based on these features we categorize them as mammals. So similarly, all those organisms with their observable physical and biological properties we can categorize. And one of the very best natural classification system is called phenetic system, right? The phenetic classification. So in phenetic classification system, it is actually a part of natural classification system. So here, based on the overall similarity, or of the overall similarity is identified through the observation of the phenotypic characteristics, right? So the phenotypic characteristics of an organisms are observed and studied, and the similarity is determined, and based on that these organisms are categorized into different groups. So you might be knowing what's a phenotype and what's a genotype. So phenotype is the expressed character, right? And genotype is the inherent root for that character. That means this is the character which is encoded on the DNA is called the genotype, and the visible character or the character which is expressed out is called the phenotype, right? So the number of attributes selected for characterization increases. That means when you increase the number of characters for the identification and characterization, then the reliability of this particular classification system also increases, right? That means when you take 10 characters for characterization, and for another organism if you take 100 characters for characterization, then the most reliable one will be the one with 100 characters, right? When the number of characters increased, the reliability of the classification also increases. And here in this classification system, each trait, that means each character, will be given equal weightage, right? Each character will be given equal weightage. So if you take the gram staining in nature, the motility, the capsule production, or the capsule presence of capsule, the synthesis of endospore, in all those characters when we analyze, and each of them will be given equal weightage, right? There is no character with a more weightage, or any character with a less weightage. So all the characters are equally considered for classification, right? So here you can use different types, like morphological and other features will be taken, and these characters are usually we analyze the characters and we'll connect them with the particular feature or particular function. Like for example, like it is very reasonable to suppose that if a bacteria has a flagella, right, if a bacteria has a flagella, then we can categorize them as a motile organism. Right? Because most of the flagellated organisms are motile in nature. So in that way, we can connect the features. Right. And in phenetic system, one of the most important thing is that this is the classification in which the organisms are compared to each other, and the overall similarity between organisms are determined. But here we have to remember that here there is no evolutionary discussion is done. The evolutionary relationships of organisms are not identified, right? There is no evolutionary pathway, there is no evolutionary distance between organisms are determined in case of phenetic system, right? So that's about phenetic classification. Hope you could follow that. So these are the different characteristics which are widely used for the study of organisms, and these data is used for the classification under phenetic system. We have a number of these are certain examples. Still, we have many others: cellular morphology, staining properties, motility, the spore production, inclusion, then the metabolic activities, the cell wall composition, membrane composition, then antibiotic sensitivity, phage typing, the serological features, and most importantly, the biochemical features. So all those features could be studied, and according to the data obtained, we can categorize organisms into different groups. All right. So this is phenotypic or phenetic classification. Now, the second one or the third category called phylogenetic classification. So here there is a phylogenetic analysis, means the evolutionary relationships of organisms are identified, right? In the phenetic system, we were not looking at the evolutionary relationships, but here in the phylogenetic system, we are much interested on the evolutionary relationships of organisms, right? So for that, when it is in higher level organism, mostly the evolutionary relationships are determined with the study of fossils and all. But in bacteria or in a prokaryotic system, the study of fossils are not practically easily possible. So here we do not look at these fossils, but instead we look at certain molecular fossils, like certain molecules which are maintaining the evolutionary relationships, or which are not much changed during evolution. So such molecules are there, like DNA and RNA sequences. So these molecules are compared between organisms in order to determine the evolutionary distance, right? So these relationships are usually illustrated in the form of a branched diagram or a tree called phylogenetic tree. You can see the phylogenetic tree representation in the diagram, and actually the three diagrams shown here are equal trees. That means these are the same tree, and they are shown in different representations. Okay, so here five different organisms are shown, each are having certain connection with each other, and they have a common ancestor also, right? So it's a graph like branched tree, and they have certain nodes, and these nodes represent taxonomic units, maybe like a species or a genus, and there are external nodes, and also at the end of the branches the organisms are represented. Okay, usually we'll be studying a present day organism, and that will be at the very end or very end of the branch, and from that organism we will look back into the ancestors, right? So this will give you an idea about how a species or a particular organism or a group evolved from a series of common ancestors. All right. So a tree may have a time scale also, right? Or the length of the branches may represent the number of molecular changes that have taken place between the two nodes, right? So length of the branch, that means length of the lines we are drawing between two organisms will represent, or between two nodes represent what? That represent the number of changes that have taken place between the two nodes. It is this actually a representation of the time scale also. Okay. And a tree may be of two types. It could be either an unrooted tree or a rooted tree. If a common ancestor is available for a particular tree, is known for a particular tree, then it is called a rooted tree. If there is no such common ancestor provided, then it is called unrooted tree. So usually for the construction of a phylogenetic tree, we have to compare and analyze various characteristics, right? The different characteristics which we have observed in the phenetic system also, phenetic classification also we will use here, right? The characters like the external morphology, the internal anatomy, behavior, biochemical pathways, and most importantly, the DNA and protein sequences, or the nucleic acid and protein sequences are also analyzed. So these features are taken, and these data is used for the construction of the phylogenetic tree, right? But these are like a hypothesis. The phylogenetic tree is like a hypothesis, but it's not a definitive answer, and they can only be as good as the data available when they are made, right? So based on the data available, we can construct a tree, and this tree, or this, when the data is changed, the tree and the relationships are also will be shifted, right? And the representation, you can have different representation, just like the image shown in the upper side. Both the trees are the same, even though the representation, the way we represent it is different, right? And from the tree, by looking and analyzing at a tree, we can identify which is the most common ancestor of a particular group of organism, and which is the common ancestor for a particular, for a cluster of organism, and which is the distantly related organism, and all. So different relationships can be identified by looking and analyzing at a tree. So during evolution, what actually happens is that there is a concept by Darwin, that descent with modification, right? So there will be the descendants, or the organisms which evolve with a modification usually. So the evolution occurs with a change. So such traits are called novel traits. So there is an evolution which occurred by modification of an existing trait. So because of that, we can look at the modification. So there was a character in the ancestor, and that character has modified in the first descendant, and in the second descendant there is little more modification, and in the third descendant there is little more modification. So in that way, a character is descended, or there is a modified character. So we will have a particular organism with a particular feature of that character, and we will look back what changes are there, what difference is there between the ancestors, right? So such traits are called novel traits. All right. And also there are certain other traits called derived traits, which are not modified, but they are acquired, or they are adopted during their growth, right? So these are developed or derived traits. So these two traits will be taken for consideration when the evolutionary tree is constructed. Usually, for the most important and significant character we use to construct a phylogenetic tree is the comparison of molecular sequences. And for molecular sequence, we use molecules like rRNA, DNA, and proteins, right? So these rRNA molecules and DNA sequences are very much conserved sequences, right? So within these regions there are certain homologous sequences, and these homologous sequences will not change during evolution, right? So there are certain homologous sequences in organisms. That means there is organism, the first organism, from that first organism a second organism evolved, from that second a third organism evolved. And when you analyze the molecular sequence of an RNA or rRNA or DNA, there are certain regions within the rRNA of the three organisms, and these homologous region will not change during evolution. Even the organism 1, 2 and 3 will have the same type of sequences, right? Or there will be very slight deviation between these two organisms compared to other sequences. So such sequences are what we call the signature sequence. So such sequences are analyzed, right? And based on that we can determine the evolutionary distance. So how much these homologous sequences are different from each other. Based on that, you can find out the evolutionary distance. Right, so it is given there a quantitative indication of the number of positions that vary in the sequence of macromolecules. Right, so these changes are also being represented as an association coefficient between organisms, which is called SAB value. So SAB values are association coefficients which are obtained by comparing the sequence of rRNA molecule, and this give you relationships or the relatedness of organisms based on the molecular sequences. So, as already said, these oligonucleotide signature sequences are the specific nucleotide sequences that occur in most or all members of a particular phylogenetic group. Right. And this SAB value, when you talk about SAB value, this is a value that give you an idea how much two organisms are related. Right, and the value of SAB lies in between 0 and 1. 1 is the maximum value and 0 is the minimum value. So if there is a value 1, means these two organisms are identical. Right, if it is 0.5, they are 50% similar. So in that way you can use this SAB value for the characterization and identification of organisms. So here you can see a kind of file, a kind of signature sequence. You can see a position in the rRNA at 47, 53, 570, 812, 906, etc. So these are certain positions which are highly conserved. Right, and the consensus sequence is also given. This is the conserved sequence in the ancestor. And in certain group of organism there is a change. The plus indicates the same base is there. Right, that means in gamma proteobacteria the C is there, cyanobacteria it is C, but in spirochete it is not C but instead it is U. So in that way you can analyze. So any change in this particular position is determined, or that will affect in the calculation of SAB value. All right. So this is a phylogenetic tree which is constructed by means of the phylogenetic analysis, or by means of usually rRNA studies. And finally, we have an addition to phylogenetic analysis. It is called parsimony analysis. And actually this is when we look at different characteristics, and when we look at different changes in the sequences, basically in phylogenetic analysis we are looking at a change in the molecular sequences, right? So when we take two organisms, there will be number of changes. So out of the number of, or out of the various changes, there will be one particular change which ultimately changes this organism, right? So in parsimony analysis, actually it represents, or it is presumed that evolutionary change occurs along the shortest pathway with the fewest changes, right? So there will be a certain particular changes which ultimately helps in the conversion of organism A to organism B. So that will be analyzed. So that will be taken into consideration in case of parsimony analysis, right? So this kind of analysis is called parsimony analysis. This is actually a part of phylogenetic discussion. So hope you follow these phenetic system and phylogenetic system. So we wind up this part of presentation here, and thank you for listening.